Hello, welcome to the demola, and today's lesson we are going to be doing carbohydrates. Now we're getting more into the diet part of topic 1, so no time to waste, let's get into it.
Okay, so to get started with this topic, we just need to focus on some definitions. So, we have monosaccharides, disaccharides, and polysaccharides. A saccharide just means sugar molecule, right? So we put mono, that means we're talking about one sugar molecule. As you can see here, we've got glucose, fructose, galactose. In terms of your exam, don't worry too much about the structure of fructose or galactose. You just want to know glucose. So glucose has two forms. You have alpha glucose and beta glucose. As for topic 1, we're going to focus on alpha glucose. Beta glucose is going to come in topic 4. Now, di 2. So, you need to know your disaccharides. There's 3 of them we need to know. Maltose, sucrose, and lactose. Now, you need to know what molecules these are made up of because they can come in the exam. So, maltose is glucose and glucose. Sucrose is glucose and fructose. Lactose is glucose and galactose. Now, you've drawn a molecule of maltose there. Just because, remember, if they want you to know how to draw alpha glucose, they can easily ask you to draw maltose in an exam, yeah? So just make sure that you're drawing this bond here. We're going to talk about it a bit more. This bond here is called a glycosidic bond. Glycosidic bond. So just be aware that we're going to talk about it more a bit later. And then your poly means it's a polymer. You got thousands of these sugar molecules. And the ones that we really want to talk about are glycogen, starch, and cellulose. So we're going to get more into them individually later. But glycogen is a polymer of alpha glucose. Starch is a polymer, again of alpha glucose, but made up of amylose and amylopectin. And cellulose is made up of beta glucose. Again, something we're going to focus on in topic 4. Okay, so like I said, we want to go on a deep dive of these different sugars, right? So starch, made up of 2 polysaccharides, amylose and amylopectin. Guys, so important that you know that starch is made up of 2 different polysaccharides, but then also that you know some features of those polysaccharides. So, amylose is unbranched, only has alpha-1,4 glycosidic bonds. We're going to explain that in a second. Amylopectin, branched, has alpha-1,4 and alpha-1,6 glycosidic bonds. And in terms of the composition, about 10 to 30% is going to be amylose, while 70 to 90% is going to be amylopectin. Just a nice fact to know, not too important, but just a nice fact to know. Starch is going to be found in foods, mainly plants. You're going to find starch in food, and it just provides the body with glucose for energy. So as we know, glucose is going to be really important for respiration, for example. That's how we're going to make ATP so that all these metabolic processes can happen in the body. So if you look at this molecule here, I've attempted to draw a bit of a polysaccharide, but what I really want you to focus on is this bond in green. We'll put the highlight on. This bond in green is called an alpha-1,4 glycosidic bond. So these are the straight unbranched ones. Whereas the branching comes when you deal with this in orange, the alpha-1,6 glycosidic bond. So you can see how both of those branches allow you to join glucose molecules or to join your sugar molecules together to make either a branch or to make a straight chain. Yeah? I hope that makes sense. And that also helps you to identify these bonds. Now, if you talk about glycogen for a second, glycogen, highly branched. You really want to mention that in the exam. Highly branched alpha glucose. So it's a highly branched polymer of alpha glucose with alpha-1,4 and alpha-1,6 glycosidic bonds. Really important in terms of storing energy inside of your mammals. And it's able to release energy rapid due to those alpha-1,6 bonds. So these alpha-1,6 glycosidic bonds here, they kind of make these 3 ends. So if we draw a dot in red, kind of like here, and that's why your enzyme is going to want to act. Yeah, those are your 3 or terminal ends. Now, both glycogen and starch have really important features. Mainly, we're going to be talking about glycogen just because it's an energy storage molecule. But I want you to mention that it's compact, it's branched, and insoluble. And like I always say, we don't mention a feature without saying the benefit of that. So compact, it stores more glucose in a smaller space. Branched, it has more for your terminal ends for your enzymes to act on to release energy more quickly. And insoluble, it does not affect the osmotic pressure. So it won't have any impact on osmosis in the body. And those are kind of your features of glycogen and its importance. Okay, something else I think is pretty important to mention is about condensation and hydrolysis reactions. Yeah. So in condensation reactions, it's a reaction in which a bond is formed releasing water. Yeah. So quick recall, glucose plus glucose is equal to what? We're going to leave you to answer that one. If you need a recap, scroll back in the video. Now, you can see here, I've drawn my 2 glucose molecules. they join together to make this disaccharide. But in that process, they also release water. Yeah. So you can count up the oxygens, you can count up the hydrogens. You're going to see that it releases a water molecule in this condensation reaction. But you formed a bond. You formed this bond here. So that's why it's a condensation reaction. Yeah. Now, hydrolysis is the opposite. It's a reaction in which a bond is broken using water. So again, we get this disaccharide with its glycosidic bond. We need to input water. And when we input water, we actually get that, and apologies, I haven't drawn it on, times 2. We get 2 of those glucose molecules. Yeah. And I just thought it was nice to mention amylase is a really important enzyme involved in hydrolysis reaction. So amylase is normally used to break down starch, breaks down a lot of the wonderful glycosidic bonds in starch. And that's a really good one to remember for your exams. Okay, so it's question time. Just 2 for today. Read them, have a go. I'll zoom in a bit so you can see it a bit more clearly. But yeah, read those, have a go, and then we'll get into the answers. Okay, with biology, so important in terms of your terminology in the question, right? This first question literally just asked you to describe condensation reaction because it was saying that you wrote 2 alpha glucose, you joined them together to form maltose, yeah? So just remember in the exam, make sure you say condensation. Really important, easy marks to do that. And then you can say that you're forming water, you're making water because it's a condensation reaction, right? So you can say formed, released, either way they'll give you the marks, right? But that is really important to mention when we're talking about condensation reactions. Now, the next question, it was kind of asking for features of glycogen, right? And guys, I'll always say this. You can see why we reinforce saying a feature and then saying its benefit, because this mark scheme is literally doing the same thing. Branched, rapidly hydrolyzed, yeah? Or you can also have broken down quicker. I would kind of favor saying hydrolyzed just because it's more technical, yeah? Compact, stores more energy or glucose, yeah? Insoluble, doesn't affect the water potential, doesn't affect the osmotic potential. Molecules too large to diffuse through the cell surface membrane is just another one. So because glycogen is quite a large molecule, it can't get out of the cells. So the only way to get that glucose out of the cells is if you hydrolyze it. Yeah, you can if you hydrolyze glycogen, then you can get the glucose out of the cell. If not, glycogen is going to stay where it is, making it really good for energy storage as well. So that's kind of an additional point you could say. But I guarantee you, if you stick to our 3 that we mentioned, you're picking up marks in the exam. Okay, so now it's time for the A-star tips. Now, what do we want to do? Guys, be really confident in drawing alpha glucose, because if they ask you in the exam, that's an easy mark to pick up, yeah? You also need to know the bonds, right? We've only mentioned glycosidic bonds because they're the only one that's been relevant now. So I'd say make sure that for now, you focus on this. But if you're a bit more further in your A-level and AS level, you're going to know peptide bonds are in proteins, ester bonds are in lipids, phosphodiester bonds are in nucleic acids. So you just need to be aware. You need to be able to name the bond when it's in a molecule. Please do not forget which bond belongs to each molecule. Yeah. Again, like I said, you link your features to the benefit. You saw it in the mark scheme. You saw it in the explanation earlier. That's the only way you're going to pick up marks, unless it just says state a feature. Yeah. That's the only way that you shouldn't then do this. But if any other way that you're mentioning features of anything, it can be carbohydrates, it can be DNA, for example. Make sure that you're saying the feature and then you're also saying why that's important, yeah? Cool. What do we not want to do? Don't confuse alpha glucose with beta glucose. If you go back to the very start of the video, we did draw out both, yeah? You'll just see that the OH group on the 4th carbon, or the one furthest to the right, you'll see that it's flipped in beta glucose, yeah? Just make sure that you don't forget that, especially when you get to topic 4, because again, really easy marks to pick up. Please do not confuse starch with glycogen. I know that a lot of the features are similar because they're both polysaccharides of alpha glucose, but you remember in your starch, you've got 2 polysaccharides, yeah? Just 2 different ones, amylose, amylopectin. So just remember that. And then also, maybe this is just me, maybe it's just me, but I used to think sucrose was a monosaccharide and fructose was a disaccharide. So I find them quite easy to confuse. So just be aware that sucrose is the disaccharide and then fructose is the monosaccharide. I don't know if I'm the only one who struggles with that, but yeah, cool. That wraps up the video for today. I hope it was really helpful. We'll zoom out a bit more. I hope it was really helpful in terms of helping you to understand this topic of carbohydrates as we explore more about biological molecules. If you found it useful, please like the video, subscribe. Also, follow us on the TikTok and the Instagram, both at demola. Any questions, leave them in the comments below, and I'll catch you in the next one. Goodbye for now.